In this activity, we're going to start by looking at onion epidermal cells. This is the skin on the surface of one of the inner pieces of onion, not the dry flaky part on the outside. This is a great first sample because the cells are large, flat, and easy to see, which makes them ideal for practicing observation and drawing skills. These slides have been stained with Lugol solution, which helps certain structures, especially the nucleus, stand out more clearly under the microscope. An onion bulb is made of thick, modified leaves that store nutrients for the plant. The outermost layer of these leaves is the epidermis, which functions mainly in protection. Since onions grow underground, these tissues are not exposed to light. Keep that in mind as we move into the pre-lab question. You'll want to think about how light exposure relates to cell function. Before we look at the cells, pause the video and answer this pre-lab question in your lab notebook. Don't worry about being right or wrong here. I'm more interested in your reasoning. Think about what chloroplasts do and whether this tissue would need them. Go ahead and pause now, and then come back when you're ready. Here's what the onion epidermis looks like under the lowest power. When you're scanning at this magnification, you're not trying to identify organelles yet. Instead, focus on the overall layout. Notice the shape of the cells. Are they round or rectangular? How tightly packed are they? Do they look like bricks in a wall? Pause the video and make a quick rough sketch of what you see at low power. This does not need to be detailed, just capture the general shape and arrangement of the cells. Now we've switched to high power, where individual cells are easier to examine. As you look at this image, start asking yourself, where's the outer boundary of the cell? Do you see a darker structure inside the cell? Is there a large, empty-looking area taking up most of the space? At this point, resist the urge to label everything right away. Just observe carefully and think about what each structure might be. Now let's confirm what we're seeing. The thick outer boundary is the cell wall, which provides structure and support. The cytoplasm is the material inside the cell where organelles are suspended. It often looks slightly grainy. The nucleus appears darker and may be off to one side. And inside it, you may see one or more darker spots called the nucleolus, which is involved in ribosome production. The central vacuole takes up most of the cell's interior. You might not see its membrane clearly, but you can infer its presence by the large, empty-looking space. Now it's your turn to complete the formal drawing for this activity. Draw one onion epidermal cell and label the required structures listed on the slide. Your drawing should be neat, proportional, and clearly labeled, but it does not need to be artistic. Take your time with this step. Accurate drawings are an important part of learning how to interpret microscopic structures. Pause the video here and resume when you're finished. The onion epidermis is a protective tissue made up of tightly packed rectangular cells that fit together like bricks in a wall. These cells have a rigid cell wall and a large central vacuole, which help maintain structure and support. Because onion bulbs grow underground and are not exposed to light, their epidermal cells do not contain chloroplasts. This makes sense because photosynthesis is not their primary function. This activity demonstrates an important theme you'll see throughout this lab. Structure supports function. As we move on to other tissues, you'll continue to compare how differences in cell shape, packing, and organelles reflect the specific role each tissue plays in the plant.